The overall goal of this assay is to measure the cellular degradation rates of misfolded proteins.
Misfolded proteins are associated with many neurodegenerative diseases. This assay helps to investigate the cellular protein quality control systems that protect us against aberrant proteins. Misfolding-prone proteins may exist in different forms in cells. By combining cell fractionation with the cycloheximide chase, the main advantage of this technique is to specifically measure the half-life of the misfolded species.
We use two model proteins as examples, a highly aggregation-prone polyglutamate protein, ataxin-1 82Q, and a conformationally unstable nuclear luciferase mutant. The protocol can also be applied to other misfolded protein measurements.
To carry out a degradation assay for ataxin-1 82Q GFP, plate approximately 3 × 10⁵ HeLa cells in 35 mm plates with DMEM medium and 10% FBS. Incubate the cells overnight so that they reach 40 to 60% confluence at the time of transfection. 4 to 5 hours after transfecting the cells with ataxin-1 82Q GFP PRK5, under a fluorescence microscope, use an excitation wavelength of 450 to 490 nanometers to examine the live cells for GFP expression in the nuclei, characterized by the presence of diffused as well as small speckles of GFP signals. Just before cycloheximide treatment, harvest 1 plate of cells by removing the medium and using 3 milliliters of ice-cold PBS